Everyone, can everyone see the screen? So shall I begin? As we all have already studied the general properties of virus, now let us come to the practical aspect and brush upon the general properties of viruses such. Viruses do not fall strictly into the category of unicellular organisms, as they do not possess cellular organization. So, defining that there are some few characteristics of the virus, that is, they have no cellular organization, they contain only one type of nucleic acid, either DNA or RNA, but never both. They are obligate intracellular parasites. They lack the enzymes which are necessary for protein and nucleic acid synthesis. Multiplication is by complex process and not by binary fission. They are antibacterial antibiotics. They are sensitive to interferons. Coming to morphology of the virus, the size of the virus, the extra particle is called as a virion. I repeat, it is a viva question. The extracellular infectious virus particle is called as a virion. The viruses are smaller than bacteria, varying from size of 20 nanometer to 300 nanometer. The viruses are much smaller than the bacteria. The small size of this and the filterability, that is, ability to pass through the filters that can hold back the bacteria, has led to the recognition of them as a separate class of infectious agents. Hence, they were for a time known as filterable viruses. Now, let us all just look upon what are elementary bodies. Elementary bodies are also called as inclusion bodies. They are nuclear or cytoplasmic aggregates of stable substances which are usually protein in nature. They represent a site of viral multiplication in a bacteria or a eukaryotic cell and usually consist of viral capsid proteins. Coming to the structure of virus, there are three kinds of symmetry which are encountered in the capsid. One is the cubical or the icosahedral. Next is the helical. Next is the complex. So there are three kinds of symmetry which are being encountered in the capsid. There are some viruses which are enveloped virus, some are non-enveloped viruses. The virions may be enveloped or non-enveloped, or you can also call it as naked virus. The envelope or the outer covering is derived from the host cell membrane when the progeny virus is released by budding. This envelope is made of it is lipoprotein in nature, and the lipid is largely of host cell origin. As I already told you, some are enveloped and some are non-enveloped, or also you can call it as naked virus. Next is coming to fibrils may be present in some of the viruses. The shape varies in different groups of the viruses. It varies among different groups of viruses. Most of the animal viruses are roughly spherical. Some are irregular and pleomorphic. The rabies virus, as you all know, is a bullet-shaped virus. The Ebola virus, filamentous, and the pox virus is brick-shaped. The tobacco mosaic virus is rod-shaped. So, different types of viruses have different types of shapes. So, this is a structure of a virus which is helical in structure, which was already covered and showed you during your introductory class of virology. The animal virus structure. Next is coming to the chemical properties of virus. The virus contains only one type of nucleic acid, as I already mentioned in my second slide. The virus contains only one type of nucleic acid, either DNA or RNA. The capsid is usually protein in nature, and they determine the antigenic specificity of the virus. Coming to the resistance of the viruses, with a few exceptions, some of the viruses are heat labile. They are inactivated within seconds at 56 degrees centigrade. Some of the viruses, they get inactivated at 37 degrees centigrade, and some at 4 degrees centigrade. The viruses are inactivated by sunlight, UV rays and ionizing radiation. They are more resistant than bacteria to chemical disinfectants, and probably because they lack enzymes. The phenolic disinfectants are only weakly virucidal. Formaldehyde and beta-propiolactone are actively virucidal, and that is why they are commonly employed for the preparation of killed viral vaccines. Classification of virus is based on the type of nucleic acid present, the homology of the nucleic acid, the type of symmetry of the nucleocapsid, the size, the shape, and the presence or absence of the envelope. This is just to brush upon some of the DNA viruses and the RNA viruses classification. It has already been taught to you. I've just put it up for just to clear. Okay, DNA viruses, it can be single-stranded or double-stranded. Okay, in double-stranded we have envelope, non-envelope. Now we have circular and the linear DNA or the DNA, and then I have given the examples for each of them. Okay, so for envelope we have the herpes virus, HepaDNA virus, then the Pacova virus, adenovirus, parvovirus, pox virus. Okay. So these are the list of viruses which contain the RNA. You can go through it later. Now coming to the laboratory diagnosis. Coming to the laboratory diagnosis of viral infections. So that mainly consists of collection of the samples, transport, cultivation, method of diagnosis. So sample collection. The appropriate specimen should be collected from the patient. It should be preserved and transported to the laboratory in the proper manner along with the pertinent history of the patient, that is the clinical history as well as the epidemiological history. As you are all aware of coronavirus at present, you can see why the epidemiological information is also important. Coming to the coming to the sample collection. So the type of sample to be collected depends upon the clinical correlation. So you can collect nasal washing, throat swab, nasal swab, sputum. Then you can go for a rectal swab in case a patient has a maculopapular rash, suspecting, okay, that is clinically if you are suspecting any of the viruses, then you have to go accordingly for the sample collection. Vesical fluids, scraping or a swab. In case of CNS infections, you can go for the CSF sample or a biopsy. In case of urinary tract infection, you can collect urine. In bloodborne infections, you can collect blood. Now, the viral transport media can be used to transport the sample. Coming to direct microscopy, coming to the method of diagnosis, that is direct microscopy. Then isolation of the virus, or you can say cultivation, then antigen detection, antibody detection, and the nucleic acid detection. So, coming to the microscopy, detection of the virus by electron microscope is being used increasingly, but in some disease it is used to be the only diagnostic method, like for example in some of the viral diarrheas. Now this is a picture showing the image of electron microscope showing the rabies virus. Note the bullet shaped virus. It is mainly employed in demonstration of the virus particles from the clinical samples, and where cultivation is a little difficult and immunoelectron microscopy is more sensitive. Detection as it is used to be the only diagnostic method. Example, viral diarrhea. In Nipah viruses, immunoelectron microscopy has been used, and it is a specific antibody is added to react with the viral antigen, and then it is visualized by electron microscope. The demonstration of the inclusion body, example Negri body bicellular stain, is a routine diagnostic method for rabies infection. So next is coming to fluorescent microscope. Fluorescent microscope can be used for the microscopic diagnosis of rabies by fluorescent antibody technique. So it can be used in various infections like rabies, paramyxovirus, orthomyxovirus, adeno, and herpes virus. This image shows you the structure, the image of a fluorescent microscope. Coming to inclusion bodies. So, as I already told you, these are these are also called as the elementary bodies. They are the most important characteristic histological feature in virus infected cells. Inclusion bodies are structures with distinct shape, size, location, and staining properties that can be demonstrated in virus infected cells under the light microscope. They may be situated in the cytoplasm, as example pox virus, or it can be situated in the nucleus, example the herpes virus, or both, example the measles virus. They are generally acidophilic and can be seen as pink structures when they are stained with Giemsa method or with eosinophil methylene blue. Some virus, for example adenovirus, they form basophilic inclusions. Demonstration of the inclusion body helps in the diagnosis of some viral infections, and the presence of intracytoplasmic eosinophilic inclusion, that is Negri bodies, in the brain cell of animals justifies the presumptive diagnosis of rabies. Next is the vaccinia. Infected cells, they show rather small multiple inclusions and are called as Gorneri bodies. Larger inclusions which are also called as a Bollinger bodies, are seen in fowl pox. Inclusion bodies in Molluscum contagiosum are also called as a molluscum body and are very large, 20 to 30 micrometer, and can be readily seen under low power microscope. The intranuclear inclusion bodies are further classified into two types, that is Cowdery type A and Cowdery type B. Cowdery type A inclusions are of variable size and they have a granular appearance. Example is the herpes virus and the yellow fever virus. While Cowdery type B inclusions are more circumscribed and are often multiple. Examples, adenovirus and the poliovirus. You can see the intracytoplasmic inclusion bodies. Next is coming to the cultivation. Cultivation of the viruses can be done by inoculating it into the animals, by inoculating into the embryonated egg, or by inoculating into the cell culture. So first is coming to the animal inoculation. The most widely used animal is the suckling mice. There are different types of routes which can be employed, that is intracerebral, subcutaneous, intraperitoneal, intranasal. So observe the animal for the sign of the disease or the death, and then the tissue is tested for the presence of virus by neutralization test or demonstration of the inclusion bodies. So next is the inoculation into the embryonated egg. So here you can see the embryonated egg. You can see different layers of the egg. So we have the yolk sac, the air sac, the amniotic cavity, the allantoic cavity, the shell. So the embryonated hen's egg was first used for the cultivation of virus by Good's Pasteur, and this method was further developed by Burnett. As I already told you, it offers several sites for the cultivation of the virus. Inoculation on the chorioallantoic membrane produces some pox, that is visible lesions. Different viruses have different pox morphology. Under optimal conditions, each infectious virus particle can form one pox. So the pox counting therefore can be used for the assay of pox forming virus such as the variola or the vaccinia. Next is inoculation into the allantoic cavity. It provides a rich yield of influenza and the paramyxovirus. Next is inoculation into the amniotic sac for the isolation of influenza virus. Next, the yolk sac inoculation for the cultivation of some viruses and chlamydia as well as rickettsia. Allantoic inoculation for growing the influenza virus for vaccine production. The other chick embryo vaccines in routine use are the yellow fever, that is the 17D strain, and the rabies, that is the Flurry strain vaccines. The duck eggs are bigger and they have longer incubation period than the hen's egg. Therefore, it provides a better yield of rabies virus, and that is why it is used for the preparation of the inactivated non-neural rabies vaccine. So this is just a summary of which is the site and which is the virus which is most commonly cultivable. So now we have different types of viral assays called as a assay of infectivity. There are two types. One is quantitative assay and one more is a quantile assay. Quantitative assay is also called as a Clark. It is again classified into two, that is Clark assay and Fock assay. So what is Clark assay? This in this, a viral suspension is added to a monolayer of cultured cells in a bottle or a petri dish, and after allowing time for absorption, the medium is removed and replaced with a solid agar gel to ensure that the spread of progeny virions is confined to the immediate vicinity of the infected cell. In this system, each infectious virus particle gives rise to a localized focus of infected cells that can be seen with the unaided eye. Such foci are known as plaques, and each plaque indicates as an infectious virus particle. Some viruses which are transmitted directly from cell to cell, example in herpes virus, they may form plaques even without an agar overlay. Next is coming to the Fock assay. Each pock on the chorial allantoic membrane arises from a single virus particle which is counted later on, which represents the number of virus present in the inoculum. It is called as a Fock assay. Next is coming to the quantile assay. So, what is quantile assay? Using serial dilutions of the virus suspension and with the aid of statistical methods, reasonably accurate estimates of the infectivity can be obtained. Quantile assay of infectivity can be carried out in animals, eggs or tissue culture. Examples of endpoints used for infectivity titration are the death of the animal, production of hemagglutinin in the allantoic fluid, or the appearance of the cytopathogenic effect in the cell culture. The titer is usually expressed as 50% infectious dose per ml, which indicates highest dilution of the inoculum that would produce an effect in 50% of the animals, eggs or cell culture inoculated. ID50 is calculated by the application of statistical methods. Now, next is coming to tissue culture. So, in tissue culture, we have different types, that is organ culture, plant culture, and we have the cell culture. Organ culture. Small bits of organs can be maintained for days, weeks, preserving their original architecture and function. It is useful for isolation of some viruses which appear to be highly specialized parasites of certain organs. So, the classic example here is a tracheal ring organ culture for the isolation of coronavirus. It is one of the best example here. Next is coming to the cell culture. We have primary cell culture, diploid cell strain, and the continuous cell line. So, coming to the primary cell culture, there are normal cells which are freshly taken from the body and they are cultured and are capable of only limited growth in culture and cannot be maintained in serial culture. So, common examples of primary cell culture are monkey kidney, human embryonic kidney, human amnion, and sheep embryo cell culture. The primary cell cultures are useful for isolation of the viruses and their cultivation for vaccine production. Diploid cell strains. These are cells of a single type. Remember here, these are cells of a single type that retain the original diploid chromosome number and the karyotype during the serial subcultivation for a limited number of times. So, here, after about 50 serial passages, they undergo senescence. The diploid strains develop from the human fibroblasts. They are susceptible to a wide range of viruses and are useful for the isolation of fastidious pathogens. They are also used for the production of viral vaccines. Next is coming to the continuous cell line. These are cells of a single type and they are usually derived from cancer cells that are capable of continuous serial cultivation indefinitely. So, the standard cell lines derived from human cancer examples such as HeLa, Hep2 and the KB cell lines have been used in the laboratories throughout the world for many years. These cell lines can be maintained by serial subcultivation or they can be stored in cold, that is -70°C for use whenever necessary. So some cell lines are now permitted to be used for vaccine manufacture. Example, the Viro cells for rabies vaccine production. Now, already you've grown the virus, so how to detect the growth? Detection of the growth in the infected tissue is seen as inclusions. So, detection of the virus growth in the cell culture can be detected by following methods, that is cytopathic effect, metabolic inhibition, then heme adsorption, interference, transformation, immunofluorescence, electron microscopy and detection of the enzymes. So coming to cytopathic effects. Cytopathic effect. Many viruses cause morphological changes in the cultured cells in which they grow. These changes can be observed by microscopic examination and are called as cytopathic effects. And viruses causing this are called as a cytopathogenic viruses. Now we have different types of formations, like we have got example is a syncytium formation, example is measles and RSV. Here the neighboring cells fuse together to form multinucleated giant cells. Then we have the cell necrosis. We have the cell necrosis and the lysis of the cell occurs in case of enterovirus. Rounding of the cell occurs in case of picornavirus. Then discrete focal degeneration occurs in herpes virus. You can see here there are few images which are causing which is mainly by having the cytopathic effect by enterovirus. There is cell necrosis by enterovirus. You can see here there is rounding of the cell caused by the poliovirus. Here, sorry. Here again you can see the rounding of the cells by the poliovirus. So now, next is, we'll go to, just a sec, let me have some water. Okay, coming back to the continuation of the cytopathic effects. So heme agglutination. What is heme agglutination? It is a convenient method of detection and assay of the influenza virus. So when the red cells are added to serial dilutions of viral suspension, the highest dilution that produces heme agglutination provides the heme agglutination titer. This test can be carried out in test tubes or in plastic trays. Red cells which are not agglutinated, they settle at the bottom in the form of a button, while the agglutinated cells, they can spread in a form of a shield. So here you can see the serial dilutions and you can see both the button formation as well as a shield pattern. Coming to the ELISA, that is enzyme-linked immunosorbent assay. So you all know the availability of molecular methods has transformed the diagnosis of viral disease, enlarging the scope as well as the sensitivity and specificity of such tests. In situ hybridization using nucleic acid probe have been used to diagnose the human papillomavirus. Nucleic acid amplification techniques like PCR, reverse transcriptase, PCR and the real-time PCR have increased the sensitivity of the diagnostic assay. Following PCR, DNA or RNA sequencing of the product also can be done.